My husband works over there. Thank you. Did you pick them? Oh, no, actually.
What?
I need that.
Okay, so if you guys want your exams, they're up here. Come and grab them. Go over what you missed. Make sure you understand what you did wrong. For the most part, you guys did pretty good, considering. A lot of people. Well, I mean, look at how this class has 45 people registered in it. How often do those people come?
Okay. All right.
Okay, so we left off talking about the light reaction. You guys know that the photosynthesis is split into two parts. We have the light reaction and the dark reaction. The light reaction is called the light reaction because it occurs in light. Dark reaction is called the dark reaction because it doesn't need light. It doesn't have to occur only in the dark, it just doesn't need light. So it can occur at any time. So for the light reaction, do you remember that there's two photosystems? Which one comes first? All right, and then we have photosystem 1. There's an antenna complex. Question?
Yeah, so if the dark reaction can it just occur simultaneously with the light reaction?
Mm-hmm. Yeah, and it can occur in the absence of a light reaction. That doesn't matter.
Is it just less? Is it less efficient?
No. They do different things, right? It's like glycolysis in a Krebs cycle, or whatever. So in photosystem 2, we have the antenna complex. Remember the chlorophyll B's and the carotenoids focus light energy on the reaction center chlorophyll, the chlorophyll A. Do you remember this one is called P680? Remember why that's called P680?
Because that's where the inverts the most light.
Right, it works best at 680 nanometers. So we have light being focused on the magnesium in the reaction center, the chlorophyll A. How many electrons in magnesium? How many free electrons in its outer shell? So when we put energy on an atom, what happens to its outer shell electrons? They get excited, and what they do a dance?
They move farther away from.
Right, they move further away from the nucleus to a higher energy level, and then that is sent down an electron transport chain to electrons. So now it's missing 2 electrons, so we need to replace those. And how do we replace those? No? We did go over this, right?
Yeah.
Okay. So water is split, and the 2 electrons from water, oxygen, and hydrogen are produced. And the 2 electrons from the water go to fill in the 2 that are missing from this magnesium. So over here we have photosystem 1. Its reaction center is P700, another antenna complex. Energy, light energy is put on that. What happens to the electrons in that magnesium? If they go where?
Higher.
Good. Now this one is missing 2 electrons, so where do we fill in those electrons? Those come from photosystem 2. We got it? Or you soon will. And then now we have the two other electrons from photosystem 1 going through another electron transport chain. The purpose of this one is not, so this one, the two electrons pump, they go through an integral membrane protein, and what do they pump out? What do they do in respiration? Right, they pump out protons. And what were those protons used for in respiration?
ATP.
They produce ATP. So how many hydrogens are pumped for 2 electrons? Good. And how many hydrogens do we need to make an ATP? 2. So from 2 electrons flowing through a single integral membrane protein, we make 2 protons, which we pump protons, which makes 1 ATP. So here, 1 ATP is made. Everybody got it? All right, so the other one, there's an electron acceptor, and respiration, what was the ultimate electron acceptor? Oxygen. Here, it's the electron carrier molecule, the one that we talked about in respiration, which is NAD, but in plants, they have a phosphate. So this converts NADP plus into NADPH. Remember how many electrons did NADH take from glucose? 2. So how many electrons are we getting here? 2. Everybody got it? So we make 1 ATP and 1 NADPH per cycle. So this is called non-cyclic electron flow because it goes directly from here to here. And in the meantime, it produces 1 ATP through the first electron transport chain, and then 1 NADPH through the second. Okay, so I told you guys, I'm pretty sure I told you this class, that we need 9 ATP and 6 NADPH to make 1/2 of the glucose, which the term for that molecule is glyceraldehyde 3-phosphate, or G3P. So this is made in an equal ratio, but this isn't. So how do we make extra ATP? Remember? Right, so the electrons can be sent back through the first electron transport chain to make extra ATP. So if it comes back, it's called cyclic electron flow. And if it goes straight on, it's called non-cyclic. So it switches back and forth. One pair of electrons goes on to NADPH, the next pair goes back. The next pair goes on to NADPH, the next pair goes back. So if you do the math, you'll end up with the 9-to-6 ratio that we see here. So we're making enough quantities to produce glucose in the dark reaction. So this is just words that follow each of those steps that we just talked about. Those videos don't work on this version of Word or PowerPoint. Okay, so we just covered respiration. You guys know that the molecule that's responsible for making ATP is called what? An electron transport. What's it called? What's the enzyme that makes ATP and electron transport? ATP synthase. Right? So remember, it spins, uses the hydrogens, it spins. So that molecule is found in mitochondria, that's where the respiration electron transport chain is, but it's also found in the thylakoid membrane of chloroplast. It's exactly the same. You could take an ATP synthase out of a mitochondria and put it in a chloroplast and it wouldn't know the difference. It would work exactly the same. So if you know ATP synthase from respiration, you know how it works in photosynthesis. And if you forgot, you might want to go back and remember that again, because it's going to be showing up again on this test. So how did it do that? How did ATP synthase work? It worked with a gradient where there are hydrogens built up on one side. They flow through, cause that thing to spin, and crush the ADP and the phosphates together. In the mitochondria, the difference in pH was a 1 pH point, so 7 and 8. I don't know if you guys remember that or not. But in the thylakoid, the pH difference is massive. The amount of protons that are pushed across the membrane is massive. The difference between the pHs is 8 and 5. So how many more protons are in the thylakoid space than in the stroma? So what's the difference between pH 5 and 8? How many more hydrogen ions? No. What's pH based on? Power of 10. So what's 10 to the third? Yeah, it's 1,000 times more hydrogen ions across that membrane. And that's 100-fold greater than the mitochondria. Because the mitochondria's difference is 10. So this is from your book. The light reaction, we just went over that. Water split, the electrons from that fill in the photosystem 2. P680 gets excited, goes through the electron transport chain, produces ATP and NADPH. Those are fed into the Calvin cycle or the dark reaction, and its job is to take carbon dioxide out of the air and make it into that sugar right there, glyceraldehyde 3-phosphate. So here's the thylakoid. Remember from chapter 6, the thylakoids in the chloroplast. So here's the membrane of the thylakoid. This is the thylakoid space, which is the inside of the thylakoid. Remember, the stroma is on the interior part of the chloroplast. And so the photosystems are in the inner membrane of the thylakoid. I might ask you that on the test. Where is the photosystems? So the electrons get excited by the light. They get moved through the electron transport chain. It pumps just like it shows you 2 protons. That gets moved on to photosystem 1, where its electrons get excited and they move on to NADPH. So in essence, we've made 1 ATP and 1 NADPH, and both of those end up going to the Calvin cycle. How do we make extra ATP? Cyclic electron flow from photosystem 1 to photosystem 2, so we can pump more protons. And then ATP synthase acts independently. This is in this order. It has to be, because we want to move electrons, right? So in order to move electrons, we need things that are progressively more electronegative. What's the most electronegative thing in this chain right here? No, oxygen. What'd you say? NADPH. This is the most electronegative. What's the least electronegative? Yeah, P680, or you could argue that water is, right, because it's transferring its electrons. All right, so the dark reaction, also known as the Calvin cycle, because it was named after a guy named, I want to say cycle, but that's a bad joke, right? So, Calvin, and basically it's similar to the Krebs cycle. So remember the Krebs cycle, hopefully. Acetyl-CoA combines with oxaloacetate and that produces citrate. So here we have a similar reaction, except it's not oxaloacetate, it's carbon dioxide. And it's not combining with, it's not acetyl-CoA, it's carbon dioxide. It's not combining with oxaloacetate, it's combining with a sugar called ribulose biphosphate. I know it's a sugar because it's called ribulose. And how do I know that's a sugar? Yeah, it's an OSE. And if it's a biphosphate, what does that tell you? There you go. So this is actually, what other word is this similar to? What other sugar has a similar name? Ribose, right. So this, how many carbons do you think is in this? How many carbons are in ribose?
5.
So how many carbons do you think are in ribulose? 5, that would be correct. The enzyme that catalyzes that reaction is called rubisco. It is the most abundant enzyme on the planet Earth. So the dark reaction of the Calvin cycle requires things to make this glyceraldehyde 3-phosphate sugar, which is actually a half of glucose. So how many carbons are in glucose? Do you guys remember? 6. So if I cut that in half, how many carbons would I have? So how many carbons in glyceraldehyde 3-phosphate you think? 3. Good. So if I want to make one of these, how many carbon dioxides do I need? 3. That's right. So we need 3 CO2s. And then I already spoiler alerted it, right? So how many ATPs?
9.
How many NADPHs? 6. So 3 CO2s, 9 ATPs, 6 NADPHs make glyceraldehyde 3-phosphate. So just to remind you, you guys have already seen this molecule. And you saw it in the last chapter. So this is the molecule that was produced when, during glycolysis, we split it in 2. Remember we made 2 3-carbon sugars? Oh, no, I don't want to cancel this. So it's right here. This is the molecule that plants produce, glyceraldehyde 3-phosphate. Remember I told you guys that plants synthesize this and then they run this reaction in reverse. So what are they ultimately making? Glucose. And then they link that together in long chains, and what do we call that? Carbs. And that's how they store their energy, just like we store our energy as fats. So, it's not like they're expecting you to come up and sneak up on them and take their carbohydrates. They're kind of storing them so that they can use them for themselves. So, how many G3Ps would I need to make a molecule of maltose? So let's think about it. What is maltose? It's a sugar. We used it in the lab. We talked about it in the lab.
It's
2 glucose molecules linked together. So how many G3Ps would I need to make a maltose? 4, exactly. So if I ask you on the test, how many ATPs do I need to make a maltose? 9 times 4 is 36, that's correct. How many carbon dioxides do I need to make a maltose? 12 is correct. 3 times 4 is 12. How many NADPH is what I need? 6 times 4 is 24, that's correct. So I'm just to let you know, remember the question that I asked you about how many, if a mitochondria is given X number of NADHs and FADH2s, how many ATPs could you make? So you guys are going to have, the final might not be cumulative, but I still expect you guys to remember what maltose is. Okay. So Calvin cycle is divided into 3 phases: carbon fixation, reduction, and then regeneration of the carbon dioxide acceptor, which is rubisco biphosphate. So it's a 5-carbon sugar. So the first step is carbon fixation. 3 carbon dioxides come in. Rubisco is the enzyme that catalyzes the reaction. It combines with ribulose biphosphate. So I just want to follow the carbon so that you guys understand that we're not creating or destroying anything. It's, you know, we're following the laws of thermodynamics. So for the dark reaction, we start with 3 ribulose biphosphates. So how many carbons in each one of these? Remember, it's ribose. So how many carbons do we have total?
15.
Good. Then we add 3 carbon dioxides, which have how many carbons in them?
1.
3 total, right? So we have a total of 18 carbons. Everybody see that? Okay. So here is the molecule that's produced from this reaction. It's called 3PG, which is phosphoglycerate. I don't care that you know that, but I want you to see that there's 6 of these, and they're 3 carbon sugars, so 6 times 3 is how many carbons? 6 times 3 is 18. So all we've done is taken 15, add 3, and we've made 18. So that we've fixed the carbon dioxide from the atmosphere into this molecule. That's why it's called carbon fixation. The next step is reduction. So what does it mean if we're reducing something? Right. You gain an electron, you reduce. So the electrons are being added, so it's reduction. Where do the electrons come from? If you have a reduction reaction, you have to have electrons, so it comes from NADPH. That's correct. Those are the electron carriers. So we have 6 of these. How many NADPHs do you think we need? 6. We have 6 of these. How many ATPs do you think we're going to add a phosphate to each one of them? 6. So we've 6 ATPs here and 6 NADPHs. That's the reduction phase. What's produced from this series of reactions, which have enzymes, but I'm not going to show you guys those, and I don't care that you know them. But what's produced is 6 G3Ps, glyceraldehyde 3-phosphate, right? This molecule right here, the half of glucose molecule. So we have to recycle this because it's a cycle. So how many carbons do we have to get back? 3 times 5 is 15. So we can't push all these back here. We won't have enough to complete the cycle. How many, if we have 18, how many can we dump out to make 15? Right, so that means how many molecules of G3P can the plant produce per turn? 1. That's it. So it's making 6, but it can only get out of it 1 because it needs the other 5 to be recycled back into RUBP, the ribulose biphosphate. So the next phase is regeneration of RUBP, which is here. Nobody knows how this works. It's kind of mysterious. You want a Nobel Prize, you can figure that one out. We do know that it requires 3 ATP, but we don't know why or how. And so there you have it. So 6 ATP here, 3 ATP here is how many? Does everybody need a nap? All right, 6 plus 3 is 9. So we have 9 ATP. How many NADPHs? 6. And how many carbon dioxides went in here? So same kind of deal as we had with respiration. I want you guys to know what comes in and what comes out. All right, so for photosystem, what goes in? What feeds the electrons? Water. What comes out? Well, it's not really that because we can do cyclic electron flow, but ATP and NADPH. And then for the dark reaction, what goes in? Carbon dioxide, right? We need the carbons to build these molecules. That's the only way you can get them. And then what comes out, or what, sorry, carbon dioxide goes in, what else? ATP and NADPH. How many carbons, dioxides? How many ATP? How many NADPH?
6.
And then what comes out? No, it's 1 G3P, which is a half of a glucose.
It seems so inefficient.
All right. So if I were to make a whole glucose, how many carbon dioxides would I need?
6.
How many ATP?
18.
How many NADPH?
12.
All right. You guys get it? This isn't going to work either. So just so you know, photosynthesis is pretty important. What would happen if the sun burned out and the plants couldn't do photosynthesis anymore? Ask the dinosaurs. Right? So we would all die. The only life that would probably still exist, it would be where? Yeah, at the bottom, because it uses what kind of process to make? More specific, please. Chemiotrophs, excellent. They don't rely on the sun. They use the chemicals from the radiation inside the core of the Earth. So on a global cell, photosynthesis is by far the most important thing. In fact, NASA has an array of satellites that sit around and watch this, the amount of photosynthesis that's being produced on a daily basis. And I'm not sure if that'll work, this is going to work, or if it'll crash work, but this is an image of, you guys know about El Nino, right? So it's a warming of the ocean that's produces increase in cyanobacteria right off the coast of South America. So cyanobacteria can do photosynthesis. So you'll see this massive explosion of photosynthesis right here in the ocean as a result of that. Which is more photosynthesis that's going on in all of the rainforests in South America. All right. Any questions about photosynthesis?
Is this happening in every gram? Or when it, is that a dumb question?
No.
So you've got the thylakoids and then the gram.
Right.
Is
So
this process happening in every single one of those?
yeah. So plants, just like us, we have multiple mitochondria per cell. Plants have multiple chloroplasts per cell, and then so we have the outer and the inner membrane, and then we have the thylakoids that are stacked together, and what you're asking is about the grana. So in each of these thylakoids, there's literally thousands, tens of thousands of these electron transport chains that are going on in that one, and that one, and that one, and that one.
Right.
All simultaneously, and all of these millions of chloroplasts that are inside the plant. So it's a massive, massive factory that's producing this. Did that answer your question?
Yeah, that's
Okay.
okay.
Any other questions?
Yeah.
So do plants have mitochondria also though?
Absolutely.
And is that just for like when they're like sleeping at night or
So,
whatever?
so
The
this
oxygen?
system, the light reaction, is sole purpose is to feed the dark reaction.
Okay,
All the ATPs that are produced here go solely to making sugars.
okay.
So the only way that they can really make energy is taking the sugars and doing what we do, glycolysis,
Okay.
Krebs cycle.
This all results in like the glucose that then goes to the mitochondria and
Right.
makes the energy or whatever.
Right.
Okay.
Exactly. Any other questions? Going once? All right. So on to the next chapter. By the way, just so you guys know, that I'm not sure that I can get through all the lecture material by in 3 lectures for the test that's scheduled the Tuesday before Thanksgiving. So I'm going to tell you now, although I don't want to move this, I'm probably going to be forced to, so you save the date for the Tuesday after Thanksgiving. That's probably when your test is going to go down.
What chapter is this on?
10, 12, and 13. Okay, so I gotta admit that although photosynthesis is super important, I'm not really a big plant fan, but I am a fan of genetics and the cellular processes. So this chapter is much more interesting to me than the previous one, although it's still very important. And, you know, the solar panels and stuff that have been developed over, you know, time have come from studying these photosystems in plants. In fact, ASU just got a $10 million grant from the Department of Energy to study photosystem 2. Why do you think that is? Why did the Department of Energy give them $10 million to study photosynthesis?
Generate energy?
Yeah, so they can increase the capacity of solar panels by understanding that. So that's called bioengineering.
Aren't they using like blue-green algae for that or something?
So blue-green algae is used for an artificial fuel. They can take the phospholipids out of it, and they can use that to make a biofuel that can power like jets and stuff like that, which is a little different than using sunlight. It's using the fats in the cells to make basically an artificial gasoline. So that's another thing that they're doing. All right, so this chapter is on the cell cycle, and just so you guys know, to remind you that all of you came from a fertilized egg. One sperm fertilized one unfertilized egg. That fertilized egg we call a zygote in biology. And so regardless if you were made in a test tube or the old-fashioned way, you still were one cell and you became a trillion cells. So how did that happen? That your cells had to divide. We talked about this. Do all of your cells have the same DNA?
Yes.
So then that means that you have to faithfully copy the DNA from that single cell a trillion times to all the other cells. And so that process is the focus of this chapter, which is cellular division. Now there's different reasons for cell division. One we just covered, which is growth. You wouldn't be able to grow if you couldn't divide your cells. But the other reason is for tissue repair. So let's say that you got cut or something happened. So you've opened up a piece of your tissue. How does that wound heal? And the way that it heals is those cells have to divide to fill in that hole. Right? So cells that were at rest are now actively dividing, and then when they touch each other, what should they stop doing? Stop dividing. What if they don't stop dividing? What do we call that? A tumor, yeah. So the cell cycle is really important in cancer biology, and just to remind you, cancer is the #2 cause of death worldwide. Heart disease. That's #3.
Medical accidents.
Medical mistakes, that's right. You got heart disease, cancer, medical mistakes, and then all other accidents combined. Car accidents, plane wrecks, people falling in their bathroom. So, tissue repair, growth, and then, so in tissue repair and growth, we want to make identical cells, right? We want to have them have the same genetic material. But in reproduction, that's different. So in reproduction, in growth and tissue repair, let's just say in humans, how many chromosomes do you guys have? 23 pairs, 46 total. So when you make a new cell, how many chromosomes do you want it to have? 46, right? Now, in reproduction, if you're a female, you're gonna make an egg, right? And if you're a male, you're gonna make a sperm. Do you want them to have 46 chromosomes? Because when they fuse together, how many would they have total? 92. And just to give you an example, one extra chromosome, one single number 21 chromosome, results in Down syndrome. So what would happen if you had two extra copies of every single one of your chromosomes? You would never be born, right? You would be what we call a spontaneous abortion or a miscarriage. That would never happen. And so what's the goal then of the chromosome numbers for the sperm and the egg? We need them to have 23, which is half the amount. So, and we have to do that through cell division, because remember, we talked about this in the first chapter, that all cells come from other cells. So the sperm and egg, they have to come from an existing cell. And through a process, it gets divided. But the goal, again, in reproduction is, when we do wound repair or growth, we want those cells to be identical, right? With the same identical genetic makeup. But do you want that for your sperm and your egg? If you had the same kids with the same person that made the same eggs and you made the same sperm genetically, what would all your kids look like?
Twins.
They'd all be the same, right? Is that conducive to life? What did we say the first day that we talked about this? What's important? Diversity, right? So the goal of creating sex cells, sperm and egg, through reproduction is we want to half the amount of DNA, and we want them to be genetically varied. Whereas when we're doing growth and wound repair, we want them to be genetically identical with the identical number of chromosomes. So there's two different processes going on. When the process, and you guys may have done in this lab already, right? Well, at least my class, I think everybody did the meiosis and mitosis lab. Yes, no? I gave you guys it as a take-home assignment, so you may not have done it yet. But you probably should before the next test. This is a strong suggestion. So mitosis, the goal of that is for making genetically identical cells. And the other one is meiosis, M-E-I, and that goal is making sex cells, sperm and egg. So everything that we talk about in this chapter is going to be similar to the next chapter, which is meiosis. So this chapter focuses on mitosis. So I just want you to think about what has to happen. We have a cell, right? It just got fertilized and you need to make another one. So what do you have to do? What does that cell have to do? okay, let's just divide it in half. Does it have the right number of chromosomes? We had 46, and we just cut it in half, so what do the new cells have? Are they the same size? If I keep cutting those in half a trillion times, what happens to them? If I have a piece of clay and I cut it into a trillion pieces, was it going to be the same size? So two very important things have to happen. What the first one is, it has to grow, right? You want it to essentially double in size. So when we talk about the cell cycle, scientists like to draw a lot of circles and cut them in parts. And so this is no different, right? It's another cycle to go along with your Krebs and your Calvin cycle. So this is the cell cycle. This one's a little easier. There's not a lot of chemistry involved. So most of your cells are in G0. This is what we call rest. So a lot of your cells aren't dividing. Like your liver cells, all of you guys look like healthy adults. So your liver cells, should your liver be getting bigger? If it is, you got a problem. You should probably go to the doctor. But at some point, it had to grow, right? So cells have to be able to control when they're in rest and when they're not. Your tissue cells, right? Smooth muscle, stuff like that. When you get the cut, the cells are in rest, right? If they kept growing, then you'd get a tumor. Not your skin cells because they're constantly growing, but underneath that. So the cells are in rest, but once you get the cut, they have to go through this process in order to make new cells. So we just talked about the first one, which is G1, which stands for growth. Some people say that means gap, but I don't believe it. Growth 1, right? So in order to go through the cycle, if you want to make an identical cell, the same size and the same makeup, it has to essentially double. And so now we want to make this one cell, we've got it to grow big enough. Can we just chop it in half now? What do we have to do next?
The nucleus has to
Well, let's just talk about so we need to copy our DNA. Right? We have 46 chromosomes. If we want 2 with 46 chromosomes, we got to double that number. So the next phase is S, or synthesis. And this is where the DNA is copied. So we got G1, S. We've talked about G2 a little bit when we talked about the lysosome. Remember, there's a checkpoint here, and it makes sure that the DNA is copied correctly. What if it isn't copied correctly? Yeah, so it undergoes apoptosis, which is basically cell suicide. But if everything's okay, then it moves on and it goes to what we call M phase. And that M can be 2 things. So M could be mitosis, and that's if we want it to be genetically identical. Or it could be meiosis if we want it to be sex cells, which we call gametes. These other cells, all cells but sex cells, are called somatic cells. And so during M phase, this is the physical process of dividing the chromosomes in the cell and dividing the cell into 2. And then what comes out of this is, if it's mitosis, we get 2 genetically identical cells with how many chromosomes in them? 46. And if it's meiosis, we get 4 genetically distinct cells with how many chromosomes? so you know their names. So that's essentially cell division, right? All right, so we've talked about genomes. You guys remember that word? I put it on the very first crossword puzzle. What does it mean?
DNA, all the letters.
The recipe book.
Oh, the genetic.
Yes, it is all the letters. So it's your genetic code. It's all the instruction set that makes you what you are. You have 3.15 essentially billion letters that tell your cells what to do. It's like a parts list. Different organisms have different sized genomes. Viruses can have small ones. There's a bacteriophage called Lambda that has about 54,000. That's its whole genome. That's all that it needs to make new bacteriophages. A phage is a fancy word for virus. There are prokaryotes like E. coli, Escherichia coli. Remember that one? That one has about 4.6 million. And then us, we're in the billion range. So genomes can vary, and it doesn't necessarily mean that because you have a bigger genome that you're more complex. Wheat actually has a bigger genome than we do. Does that mean that wheat's more complex than me? Because I'm pretty sure I could outthink a grain of wheat. Maybe not. So all of our DNA, you guys probably already called this, is organized into chromosomes, and we've talked about it. Some of you guys that have done the lab, looked at the slides, the onion slides, have seen the chromosomes under the microscope. And so, and we've talked about this before. So the chromosomes are made of not only of DNA, but of proteins, right? And together, those proteins are called histones, and together the DNA and the histones make up chromatin, and long stretches of chromatin make chromosomes. So this isn't new. We've already talked about this. What is new is that, and I've talked about the example of flying the kite, right? And talking about the spool in here. And so what I want to kind of impress on you is that this is a massive amount of information that you have to fit inside the cell that you, in the nucleus of the cell, that you can't see with the naked eye. So 7.5 feet of DNA have to fit into every single nuclei of every single cell in your body. And how is that accomplished? And it's no easy feat. So just to focus on chromosomes really quick, is that this is called a karyotype. When you take a cell and you extract the chromosomes, this is done pretty commonly whenever you want to test, like if your baby is going to have a birth defect, like an extra chromosome or something. And just so you guys know, maybe you don't know this, but the fertilized egg, the cells from the fertilized egg actually make the placenta. So cancer biologists are interested in this because it's one of the only times where different cells invade other cells. So what does that sound like if you have one set of cells going and moving to another place, and what else? It's cancer, right? So the cells from the fetus produce the placenta, and so if you want to test what the fetus DNA looks like, all you have to do is sample from the placenta, right? Or the amniotic fluid, which is the cells are sloughed off into there. So what they do is they'll take the cells and they stain them. This is a special technique called fluorescent in situ hybridization, which is a fancy word, but we all in the medical and scientific community call it FISH. Fluorescent in situ hybridization. And so you can stain the chromosomes with this technique, and they end up with a different staining pattern so you can identify them. And so this isn't how they come out, right? You don't like just watch the chromosome and they're all automatically paired together. This is all photoshopped, right? In the old days, they would take a picture of the chromosomes and then they'd have to take scissors and cut them out and like paste them on, you know, poster board and match them up and junk. We don't have to do that anymore. And then the chromosomes are numbered based on their size. So chromosome number 1 is the largest. Chromosome number 22 is the smallest, although when they made that decision, it looked like it was the smallest, but then when we sequenced the DNA, chromosome number 21 actually has less DNA in it, so it's actually technically smaller. So do you think they changed the number? Why would they? They didn't change photosystem, so why would they change this? So 21 is the smallest chromosome, but that's a mistake. And then this is an X chromosome, this is a Y chromosome. So if you're an X and a Y, do you guys know what you are?
Oh boy.
You're male. And then if you're XX, you're female. So this, you could tell if this child had Down syndrome, you would see 3 number 21 chromosomes here. So this is a normal male.
Can you get extra chromosomes somewhere else besides 21?
Yeah. So you can get an extra 18, that's survivable. 21, 13 is survivable, although not very long. Most people, this is the longest survivable extra chromosome. Number 18 and number 13, usually offspring that have that condition die within the first year or 2.
Does it have anything to do with the size as to why it's survivable?
Yeah, so the ones that have the less genes that are involved have the better chance of surviving. There are also, you can get pieces of chromosomes. So there's a symptom called cri du chat, which is French for cry of the cat, because when the kids cry, it's sort of a mew, mew sound, and that's caused by an extra piece of a chromosome 5 getting added on there. So not the whole chromosome, but just a part of it. So there's all kinds of genetic variations that you get from the. There are people that are, some of the more survivable ones are the extra sex chromosomes, because in females, if you get 2 X's, one of those, having 2 doses of X chromosome would be lethal, so you have to shut one down. So in all females, only one of the X chromosomes are active. The other one becomes inactive. And in males, they only have 1 X chromosome, so it doesn't affect them. The Y chromosome has a gene on it called SRY, which is the gene that causes you to be male. So all of us are predisposed, if everything was left to just nature, and no interference from any chromosomes, everyone would be predestined to become female. And it's only when that gene is activated that the pathway to become male is sort of evoked. So you could have someone that, and this has happened before, like, you know, you see these athletes that are getting tested for their chromosomes, and they're like, well, that looks like a guy, but it has 2 X chromosomes. But in some cases, you could have a part of the Y chromosome, that gene put on the X, so physically it would look like 2 X's, but in reality, all you need is that gene, no matter where it is, and that makes you a male. So you could also have, there are females that have 3 X chromosomes, and that doesn't matter because it just shuts down 2. You could have 100 X chromosomes and it would shut down 99. And there's lots of conditions where you have XXY, so that still leads to being a male because there's still a Y chromosome. And then there's lots of conditions where you have X and then multiple Ys. So what do you think? What do you think causes you to be male? Right, but But what does the Y chromosome make that makes you male? Testosterone, right? So what do you think would happen if you had extra Y chromosomes?
Yeah, I like testosterone now.
Right. And so what does that lead to?
Muscle, rage.
Right. And so there's studies that have been done on people that are in prison, and it turns out that in the general population of prison, there's a far higher, like a 50% higher incidence of multiple Y chromosomes than it is in the general population.
So what about, like hermaphrodites? What, is it a gene that causes that?
It can be lots of things. We'll talk a little bit about that when we get to the chapter on genetics. So I don't want you to memorize all this, but basically I want you to see that there's quite a bit going on. So you're taking DNA that's 2 nanometers wide, you're spooling it around. What are these yellow things called? Histones, excellent. And then those get condensed, they get looped around, and then coiled, and then that's just a small portion of the chromosome. So here's a typical chromosome that you might see in your book, or I probably should leave that up. Let me erase this. And chromosomes always look like this X shape, right? From what you've seen. But they're really only X shaped because they're individual chromosomes here, here, and then here, what happens to them? They get doubled, so they're actually held together by what we call a center part. What do you think center is in Greek? It's close to Spanish.
That's great.
It's very close to Spanish. Yeah. And then what is part? So that literally means center part. So that's why it looks like an X, because there's actually 2 of these being held together by a centromere. Center part.
Did you ever have 45 chromosomes?
Yes, but there's only one condition. It's called Turner syndrome, and that's a female that only has 1 X chromosome. But there's no survivable non-sex chromosomes that have 1 too few. No one has ever been born with that condition.
But if you shut it down anyway, what does it matter if you don't have that X chromosome?
What happens if you only have 1 X?
So some people with Turner syndrome, like they don't menstruate, they become sterile. Sometimes they're a little slower, for lack of a better word. But it varies, right? And we don't really understand all of this stuff. There's a lot that we know about genetics, but there's also a lot that we don't know.
In the Down syndrome, like what is it about the chromosome that gives them their facial features? Because they kind of all have the same facial features.
So I'm not sure about all the chromosomes, all of the genes on chromosome 21 and how they interact, because it can get complicated, right? I mean, if you're talking about like genes that are interacting, then you're talking about like exponential genes. So let's say you have 2 genes that interact, and so the combinations are 2, right? But then if you have 4 that interact, it's 2 to the 2nd power. And if you have 3, it's 2 to the 3rd, 2 to the 4th. So if you have, there are 3 genes that control skin color, just to give you an idea. So the number of genes that are involved in shaping a face are probably 50 or more. But there's definitely, a physical appearance that people with Down syndrome have. I just don't know enough about the genetics behind that chromosome to give you an answer.
I
guess
but there is something.
they don't have a bone right here. That's why their foreheads are very like flat.
I just learned something new. I didn't know that.
Yeah, that's how they check. Sometimes how they check to see if they double check to see if the baby has Down syndrome once it's like older.
So there's probably a set of genes that are involved in that. Developmental genetics is really complicated too. There's whole classes that are dedicated to developmental genetics. Hey, and it's not blinking, so I can turn it back on. Okay, so this is really important because it's terms that we're going to use not only here, but in the next chapter and through genetics. So you guys know that, let's just say that you have a number 1 chromosome, right? How many number 1 chromosomes do you have? 2. And where did they come from? Nobody knows where they got their chromosomes.
Each parent.
Yeah. So let's say that this is, I'll use a black marker for dad and I'll use a red marker for mom. And so when you undergo synthesis phase, just for effect, let's say that this, so mom gave you the brown-eyed gene and dad gave you the blue-eyed gene, right? So because these chromosomes are similar, but they're not exactly the same, right? This one's obviously different because it has a different gene, but it's still a number 1 chromosome. We call these homologous. Chromosomes. We got that term? Okay, so when we go in S phase, you're going to make an identical copy of this, and it gets held together by the center part, centromere. And so what's that gene going to be if it's exactly the same copy? This one on this new chromosome. That one's going to be brown too, right? It's not going to be blue. And then this one's going to get copied. And what's that gene going to be? Blue, right? So because these are genetically identical, we call them sister chromatids. So just to recap, if the chromosomes are similar, right? Like number 1 chromosomes that you got from the mom and dad are number 2. We call those homologous chromosomes. If they're genetically identical, produced by the S phase copying of them, we call them sister chromatids. So those are, if you don't learn anything else out of this, that's what you should learn, because that is critical in order to understand what's going on in meiosis and mitosis. And it's pretty easy to tell sister chromatids because they're almost always attached by a centromere. So if you see that X-shaped chromosome, you know those are sister chromatids. Because homologous chromosomes aren't attached to one another. I mean, there is an exception, and we'll talk about that later on, but for the most part, they're not.
This one over here is
Okay. So this part of the cell cycle, M phase, this chapter is on mitosis. So we're just going to focus on mitosis, and then in chapter 13 we'll do meiosis. So mitosis is divided into 5 parts. This one is called prophase, and this one is called metaphase. This one's called anaphase. This one's called telophase. And this is how it was for the longest time. There was only 4, but then somebody somewhere decided that we didn't have enough phases, so we had to put another one in here. And so this one's in between prophase and metaphase, so what do you think it's called? Yeah. Prometaphase. And then there's another part at the very end, which is considered separate, but usually people just consider that part of telophase, but since your book addresses it as separate, we'll just make it separate, and that's called cytokinesis. What's kinetics mean? Moving. In this case, it's moving the two cells apart, so it's physically separating the cells. Cyto means cell. So just to remind you, the goal of mitosis is to make genetically identical, same number of chromosomes. So those are called somatic cells. When we're talking about meiosis, what's the goal? We want half the number of chromosomes, right? And we want them to be genetically different from one another. So the goal here is to make exact copies. All right, so I've already done the cell cycle. Do you guys need me to go over this again? What's G1? Purpose of that is growth. S is copy the DNA. G2 is what's its purpose? Make sure the DNA is copied correctly. If it's not, what happens? Apoptosis. If it is, then it goes on to
Can you just call it quality assurance?
mitosis. What?
Can you just call it quality assurance?
Sure, whatever you want to call it. Just as long as you know G2 checks the DNA to make sure it's copied correctly. So you could say G2 QC, quality control. All right, and then, so this is what we're going to focus on now, is this mitosis part. So mitosis, like I said, 5 phases: prophase, prometaphase, metaphase, anaphase, telophase. Just, you know, so the whole semester we've kind of been going over the Greek and Latin roots. So do you remember what pro means? Before. So prophase is the before phase or the beginning. Meta is, any guesses? Middle. Prometa is before the ana. Remember what that is? So I'll explain why it's called anaphase, but it has to do with the movement of the chromosomes. And then the last one is telo, which means end. You guys are going to see this again because the ends of the chromosomes have a specific name. What do you think they're called? Come on. It's the end part. Telomere. That's exactly right. And those are important because they have a lot to do with cancer as well. So you'll see that coming up. All right. So prophase, the nucleoli, I'm sure you guys don't remember this, but in chapter 6 we talked about the dark spot that's found inside the nucleus, and that's the site of RNA, ribosomal RNA synthesis. So that is gone because in the chromosomes have to condense. Because remember, you have 7 feet of this DNA, right? So each chromosome, if you have 46 chromosomes, they're relatively long. So just think about it this way, and this is what I told my other class. So if you went to the gym and you threw 46 pairs of headphones, earbuds in your gym bag, just chucked them in there, ran around with them, what would happen when you wanted to take one out? They get all tangled. And the same thing happens to chromosomes, right? You don't want those things tangled up because when you pull them apart, what happens? If I just pulled apart my headphones out of the bag, what would happen? They would break. Would they work anymore? Same thing with chromosomes, right? They would break and they wouldn't work anymore. So how would you get, riddle me this, how would you get 46 pairs of headphones in your gym bag and keep them from tangling with each other? Wrap them all up. There you go. So that's what your cell does, right? It just wraps all of this. It condenses the chromosome. So in essence, the chromosomes are condensed 50,000 times. So they get 50,000 times shorter and 50,000 times wider. If you took a rubber band, oh, there's one here, and you start spinning that thing, you're condensing it, what happens to it? It gets shorter and it gets wider, right? So that's why you're able to see these things, because you can't see chromosomes unless they're condensed, and they condense here. So any of these other phases, you cannot see the chromosomes. This is called interphase. And these have to condense, because if they don't, when you start moving the chromosomes apart, they're going to tear. So the chromatin condenses. Remember, the chromatin is made of DNA and proteins called histones. There are mitotic spindles. These things, they're called centrosomes. They move apart from one another, and what they're doing is, so here's our cell, here's our nucleus, here's our chromosomes. And so the goal is that we have to attach to these and then move them apart into two new cells. So these centrosomes, they move apart and they start making these microtubules. Remember we talked about this, the cytoskeletal component that's responsible for moving chromosomes is microtubules. And you know, think about it, how hard would it be for me to open this pen if I only had one arm? Let's exclude everything, but because I can't get opposing force on it, right? I could use my mouth, but I'd have to get some sort of opposing force. I can't just go like this and open, right? So you have to use opposite force. That's why you need two of these on opposite sides. So they start to move to opposite sides to generate that force to pull these apart. Make sense? My past time. All right, let's just finish this one. So, and then the centrosomes move apart, but there's an issue here, right? What's in between the microtubules and the chromosomes? The nuclear membrane, right? And the microtubules can't go through the nuclear pores. So the nuclear membrane has to be dealt with. And so we'll talk about that next time. If you guys haven't got your test and you want it, it's up here. You can frame it or burn it and eat the ashes. It's up to you.